My name is Jim Heath, and I'm the professor of the Institute for Systems Biology here in Seattle. And I'm here today with Dr. Jingyi Ji. Jingyi was a graduate student in my group and is now working at the Allen Institute just a few blocks away. And Jingyi has just published as first author a paper from her graduate work entitled ATMAT Analysis Reveals the Association Between CD8 plus T-cell receptors, cognate antigen, and T-cell phenotype and persistence. A bit of a mouthful. Congratulations, Jingyi.
Thank you, Jim.
So this paper represents a very large-scale analysis of T-cell receptors paired to their antigen, viral antigen, paired to the actual behavior of the T-cells once they see the antigen. It involved both technology and science. So maybe, Jingyi, why do we care about CD8 plus T-cells to begin with?
Yeah, sure. CD8 T cell is also called cytotoxic T cells. One fact is that CD8 T cell response is antigen specific, which means that they recognize a short peptide from a virus fragment through a surface expressed T cell receptor called TCR. This interaction is highly specific, and once activated, they can kill the virus-infected cell. And another fact is that some of the T cell can develop memory phenotype, which means next time when they see the same antigen, they will act fast and expand and then eliminate the virus again. So in summary, CD8 T cell response is really important in adaptive immunity and in clearance and develop long-term immune protection.
Right. And so in this paper, a fairly audacious part of the paper is that you turned the entire SARS-CoV-2 proteome into a library of basically T-cell fishhooks, antigens that could capture T-cells that recognize those fragments of the SARS-CoV-2 virus. So what were the key technologies that allow you to do that?
In this paper, we were utilizing a technology called single-chain trimer technology, which allow us to produce the peptide MHC library in a really large size. So here, we are sampling the entire SARS-CoV-2 genome, and then we ended up with over 600 of peptide MHC specificities using the SCT technology.
So that's a lot of antigens.
Yeah.
And on top of that, when you did the analysis, you looked at over 60 patients at the same time.
Yeah.
So what allowed you to do that level of, I guess, highly multiplexed analysis?
Yes, so I guess that's the experimental side of our APMAT paper, is that we paralleled using the over 500 of SCTs and then also over 60 patients because we utilizing the DNA barcodes and hashtags and then utilizing each patient's genetic differences so that in the end, we have a large library of T cells that have their individual antigen specificity, T cell receptor, the RNA gene expression, and then the patient ID, allow us to dig more about the interaction.
So that's a pretty big data set. And one of the mysteries that we tried to address in this paper, I guess it's an open question, was how does the T-cell receptor gene, the actual sequence of the T-cell receptor that recognizes the antigen, how does that influence the function of the T-cell once it encounters the viral infected cells in the body? And so you put together a computational model to try to sort through that. Maybe you could say a few words on that computational model.
Yeah, sure. So we're actually focusing on the peptide amino acid sequences and also the biophysical, biochemical properties on each of the amino acids of each antigens. And we do the very similar thing for each of the T-cell receptors specific to those antigens. So then we can characterize all those large groups of peptide into 3 groups, and then all those T-cell receptors into 2 groups. Yeah, so we have a combination of peptide and TCR physical chemical properties then.
And then what you found is that, in fact, the physical chemical properties of the T-cell receptor antigen by and large dictated the T cell function.
Yeah, exactly. So when we have different peptide, TCR peptide groups, we look at the gene expression profile. We see that some of the peptide TCR pairs tends to be naive, and then some other pairs tends to be more cytotoxic.
Right. And you also found that you could predict which cells would persist over time in these patients and which ones would not.
Yeah, and I think one cool thing about our study is that we've previously published longitudinal data sets for the same set of the cohort. So although experimentally we were capturing those T cells from acute disease stage in this paper, but we got the TCR sequences, so we can track them down in our longitudinal data sets to see what T cells still persist at a later time point, even say like 2 or 3 months after disease recovery. And what we found is that the different peptide TCR pairs associated with not only the phenotype at acute disease, but also the persistence at a longer time point. Yeah.
So in other words, you could predict which T cells would kill and which T cells would remember.
Yeah.
Pretty cool. So what's next? What are the big questions that you think this technology allows you to address?
Yeah, what I can think is 2 directions. The first one is that, as we mentioned, we focus on the most prevalent HLA AO201, which is by and large a large fraction of the whole population. But we still wanted to extend the similar concept to a population level, which sampling for other HLA alleles to see what we found here is whether it's applicable to other HLA alleles. Another thing I can think about is extend our AP-MED framework into the cancer immunotherapy that you've been recently worked with.
That's correct. It's an excellent way to try to identify tumor-reactive T cells that can then be harnessed for cell-based immunotherapies, and that's exactly how we're using this technology right now.
Yeah.
Thank you, and congratulations again.
Thank you, Jim.